Oh, wonderful. Well, hi, Sanket.
Hello.
And welcome to the Cellmates podcast. We created this podcast at Multiply Labs to discuss, welcome our partners and show people how we work together, why we developed this technology.
Thank you. Glad to be here, Fred.
So the team prepared some cards for us. We don't know the content of these cards, and so we're gonna unveil the first one now and then answer this.
All right.
Okay, so let's see. Give us a quick rundown of who you are and what you do.
Right.
No corporate speak.
Hey, you want to go first?
I can start. I mean, my job is kind of like the easiest to basically define because I basically have no jobs. It's not a clearly defined job. My job is I need to deal with the worst problems.
Yeah.
But I also need to come up with the new directions. And so, for example, that's why I've been very involved in this collaboration, with the GenScript team. I need to be very involved in those, high potential, high return initiatives where, it starts from an idea. Usually, I'm the best person to interface with the decision makers on the partner organization.
Yeah.
So saying founder and CEO basically means saying nothing because it's, whatever is most important in that moment.
That's right. Yeah. Yeah. You're trying to get rid of all the fires out there.
Pretty much. While, looking for the new big directions.
Yes, exactly. Looking for strategic direction. I think I would say it's kind of the same on the GenScript side. So I'm part of the corporate development team. And basically what that means is supporting development of our organization through collaborations and strategic partnerships. So I think we are very fortunate to be working with Multiply Labs because I think it's a great synergy between expertise in cell therapy and development of the instrument, whereas you guys have the robotics expertise and the engineering expertise. So I think together we're able to create something unique.
Yeah, you're right. And I do feel we have a very similar approach. We both move very rapidly. I would say motivate our team to work very closely together because that's the only way you're gonna, get stuff done rapidly. And so you need to share that mindset. Otherwise, stuff doesn't happen.
Yep. Yeah. And I think we really believe that we all have a role in the ecosystem, and if we do a great job at creating the best reagents or the best instruments, high quality instruments, and then if you're able to robotically integrate them into your platform, I think it really creates value.
Exactly.
In the whole ecosystem.
Wonderful. So, next question.
Okay, so what's the project you're collaborating on and what makes this partnership interesting or unique? All right, I can go first.
Yes.
So, as I mentioned earlier, we're really happy to work with Multiply Labs on this collaboration. The concept was, would there be value created in automating other unit operations like cell separation, where we don't have a lot of options out there. So as some of you might know, GenScript has the Cytosync 1000 instrument, which is a cell separation device based on magnetic beads. We have the nano beads as well as the consumables for that go with the Cytosync. And Fred had this unique idea where we thought, could it be possible to robotically automate the Cytosync? And we thought that was a great idea. We thought it was really important because you see a trend in more and more automation in the manufacturing space. And specifically with cell therapy, Multiply Labs' approach to automation was really intriguing. And so that's why we really got interested in working with you.
Yeah, there was great alignment right from the start. Our approach is that we don't design new instruments, rather we teach our robotic systems to use best-in-class instruments, consumables, and reagents.
Right.
There is a huge trend and a huge push towards essentially end-to-end automation in cell therapy manufacturing and a very big unit operation for which we saw so much demand was, isolation and enrichment. It was very natural to start interfacing with the GenScript team. They have the Cytosync, they have beads, and that was exactly in the direction where we were seeing the leaders in the market asking us, do you have an automated solution for that? And that was, to us, the beginning, of the conversation with GenScript. And so the idea here is, okay, can we teach a robotic system to use the Cytosync,
right,
the consumable kit, and the beads, so that if you were a cell nothing will change, basically, or as little as possible will change. That requires really a very close collaboration between our robotics engineers and the GenScript essentially engineers. It's the same instrument, the same hardware, the same software, the same consumables and reagents, but now augmented essentially by being able to work very closely with our robotic system. And we heard many times, hey Fred, hey GenScript, Multiply Labs, can you help us automate an end-to-end process? And this is a huge step enabling that.
Yeah. Absolutely. I think I do remember, during the collaboration, one of the key kind of bottlenecks was to figure out a way for the robotic system to talk to the software of the cytosync. And I think the brainstorming we did there to find a solution was quite memorable as a almost like a model for how you could potentially automate other solutions.
It is because we cannot and do not want to modify the original instrument. It is very important to work with the team that has developed it. And I think the collaboration with the GenScript team has been so close between the engineering teams, right? Not just at the decision-making level, but also the people that actually make it happen. Our teams now know each other's, have each other's contacts, and they're used to work so closely.
Yeah.
These would have never happened this seamlessly, and would have never been so successful if the teams were not basically committed to working together so well. It was like a continuous exchange of people and ideas. It's a great model of how to automate, an instrument, but it's also a great model of how to run essentially a partnership in this industry.
I agree, totally agree. There's so many learnings from this. Okay, great.
What's the bigger value of partnership in pharma automation overall? This is really a central, why we're here, why we work together. Essentially, cell therapy and in general advanced therapies are hard. Reinventing the wheel, it's not, a particularly wise idea. It is great to partner with the best people for their areas. And so working for enrichment, isolation, consumables, reagents, working with GenScript is fantastic. They are leaders in the space. On the Multiply Labs side, we are focused on developing the robotic technology. It's really complementary, and partnerships allow you to bring the strong points together without needing necessarily to engineer the things that we're not specialized in. So for example, if Multiply Labs had to design a new reagent, right, it would take us years, and we would not be as good as the leaders in the space, right? And so it just makes sense to partner. I think it's even more important in a space like advanced bioprocesses, where even a tiny change in a process can have devastating consequences, because you never know really what happens to the cells if you change even a single consumable or reagent, let alone an instrument. So there is a huge motivation to work together, combining proven technologies.
Absolutely, yeah. And I think you see the value of partnerships, across the continuum of drug discovery and development, right? There's R&D, there's technology, manufacturing, and then there's patient access as well. So there's so many areas where you could potentially have partnerships. But I think especially when we come to cell therapy manufacturing, I think when we first started working in this space, both cost and manufacturing have been the bottlenecks for autologous cell therapy manufacturing, and potentially allogeneic too. When we looked at Multiply Labs platform, what really struck us was you could still use the same instruments, like you said. You could use the same consumables. All of these instruments have passed the regulatory checks, but at the same time, you could make the process flexible, almost giving manufacturers and pharma partners a choice for what they want to automate. And also the flexibility as well as the modular nature, right? So you can contract the process or expand the process. I thought that was really intriguing. If it wasn't for Multiply Labs, we wouldn't be able to do that. So I think that's the value that partnerships has, where both teams come together and create something bigger than each of the parts.
I'm very curious about this new question. So how has the world shifted from the old way of pharma manufacturing, and why is automation so critical now? Should I go take a first stab?
Sure,
okay, great. So, so essentially, the old way of pharma manufacturing has essentially limits of scale. I think we've seen that not just in pharma, but with all advanced technologies. They all start at a, I would say, artisanal level, where, you know, they're kind of handcrafted. You're still relying a lot on, you know, an expert putting it together. I mean, semiconductor started like that as well. Car manufacturing started like that as well, right?
Exactly.
But then at some point, when you need it to be a widely available product, that's not just for the first lucky few, right, or not niche, but like really like for millions of people, essentially, historically, you've always need to bring in, you know, more automation.
Yeah.
And, you know, I believe we are at that point right now for advanced pharmaceutical manufacturing, and I think cell therapies are just a fantastic case study, almost like the kind of like they're opening the way for everything else.
Yeah.
What we are doing right now is essentially showing, you know, a new way that's not artisanal, it's not manual, it is actually fully scalable and, you know, sustainable from an efficiency point of view.
There were so many published studies where you see that cost is a major driver that's impacting access of cell therapies to patients. A huge part of the cost of manufacturing cell therapies is labor. Automation is a way to address that, right? Because if more than 50% of the cost is labor, it's because, you know, you need trained personnel, PhDs, to actually do the manufacturing of a very time-intensive process. And so if you can reduce that with manufacturing, with better reagents, with automation, there's a way for us to reduce cost. I mean, I think there are 2 or 3 CAR T therapies now that have been approved in China, where the cost is about $120K. The US therapies are almost $350 or $400K.
Yep.
There are therapies in India that are like $60 or $70K. So it is possible to reduce the cost of these therapies through automation. And that's kind of a key driver, I feel like, for using automation to just provide medicines, these life-changing medicines, to more patients.
Yes, labor is right now a big cost. There are not enough operators. So I've never met a cell therapy organization that has told me, oh yeah, you know what? We can hire very easily manufacturing operator. That's not a problem. I've never heard this word. But even when you talk to, you know, the manufacturing operators themselves, right? You know, very highly qualified. You know, usually they're very excited to work on the development phase at the beginning, right? So our scientists in the multiple labs team are super excited, you know, to be working with the robots to develop, you know, new automation.
Right.
But then none of these people studied that much to be essentially line manufacturing operators in a clean room. So it's not just that labor is, you know, unsustainably expensive. There's also like, this is not a job people want to have. It's much more interesting to supervise automation. That's also, I think, very aligned, you know, not just with the needs of sustainability and, you know, scaling the industry, but also a kind of job that people want to have.
And somewhere they are closer to patient outcomes rather than just being in the...
Exactly. It's also nerve-wracking, honestly, like in a single process there's maybe 500-600 individual tasks. Knowing that if you miss a single one, right, it could result in a deviation or even loss of the product. And these are life-saving products. And so definitely much better to watch a robot do it from outside the clean room than it is to be the robot inside the clean room.
Potentially less error too, right, overall.
Okay.
One more.
Next.
All right. Oh, this is to both of us. Okay, to Sanket. What's one thing engineers often miss about science? I guess it's less about what they miss. You know, I'm trained as a scientist, so I would always think about growing cells and how they behave. And the first thing you realize about cells is that sometimes cells can be very unpredictable and they are not precise. So it's not X or Y kind of situation. There's a lot of gray room for how cells behave. So it's never like 0 or 100%. It's like 20/80, 20/60, you know. And so maybe that's something to consider when engineering solutions, to have that inbuilt flexibility or variability. So if you look at leukopaks, you know, every cell, the cell numbers are different, viabilities are different, every sample has different potency. I think engineers really help us make things, standardize things, which is really critical because, you know, biologists cannot do that. so that's where we would learn from engineers.
Yes, I mean, that's very true. For me, what's one misconception scientists often have about engineering? Well, so actually it's connected to this. Engineering is so repeatable. But very interestingly, when scientists work with us, typically what they notice is they're like, can the robot go faster? Can the robot go faster, right? And the answer is yes, it can. But the way the robot go faster, it's a very, very different, the chart of progress in biology and in engineering is very different. So in engineering, you have a very steady rate of progress. For example, when the robot moves slowly but get more and more robust. And then once you're sure you got it down, like that task, is very repeatable, then cranking up the speed is actually quite straightforward. So typically your 90% is getting reliable, and then the last 10% you crank up the speed by 3-4 times, and that's fine. In biology, it's sort of like non-linear, or I guess double linear, like linear and then step. It never happens. You never have cells that improve steadily and then all of a sudden improve by a lot. Typically, biology is, at least in my experience, a set of pretty much plateaus, or sometimes even going back, and then there is a step improvement. Right? And so to me, the two scientists and engineers are trying to understand each other's work, but they assume that their systems behave the same way, while the cells and the engineering system behave in very, very different ways.
Yeah, exactly. I remember once when you were showing us the robots, and I think one of the questions we had was like, can the robot move faster?
And then, when we're sure, we accelerate it, and then people get scared. Oh, Fred, the robot is moving too fast. And we're like, well, from the engineering point of view, it's actually the same.
And probably if you think about manufacturing a cell therapy, cell expansion takes so long that it doesn't matter if the robot moves slowly or not.
There's also that, yes, typically from the point of view of a cell, the robots are always, they're like the Flash. Like the cells can't realize how fast the robots are moving because a cell reacts in seconds or minutes or hours sometimes. And the robot is measured typically in seconds, sometimes milliseconds, right? That's another thing. The time scales between biology and engineering are so different. So to control the robotic arm, a kilohertz would be normal. That's 1,000 measurements per second.
Wow.
In biology, that would not have meaning.
Not be possible. So, wow. No constraints. If you could innovate anything in pharma automation together, what would it be?
Oh, that's an interesting one.
I mean, I guess making the drug development process faster would be a great way. I think protein design is a great area where you could use AI and automation to design new proteins and see what their manufacturability is.
It's true that so far we've been focused necessarily due to the demand on advanced clinical or even commercial applications, but there is a huge potential early on to use automation to facilitate maybe late preclinical, early clinical, basically manufacturing or discovery. A huge initiative right now that we're starting to work on, essentially, is it possible to auto-train the robots for a new process, right? So let's say we automated the leading instruments like we're doing now, right? Okay, now the robots can use this instrument, but there is still a bottleneck, which is how many new processes can the robot learn and how fast?
Got it.
What if we could start from just expert demonstration, right? Just video data of a scientist running a process on instruments that are of course compatible with our robots, right? And what if from that video demonstration the robotic system could auto-learn? We would not work with a few dozen, we could work with hundreds of them because all we would need to train the robots are typically video-based demonstration.
I see. I think similar to that, one of the things that we talked about in the past is probably software that can, easily connect with any instrument, right? I think some of the problems that we've faced is having different softwares and finding ways for them to connect to each other. So if there's like a standardized way for all instruments that are out there to interact with each other, it would be kind of a piece of cake for, you could take any instrument, you could have any instrument next to it in any workflow, so because they're all standardized and all know how to talk to each other. I think that would be a really powerful addition. With AI, we could, even reduce some of the process issues that take place. So I think that's something that would be that would have a great impact on our space.
Yeah, absolutely.
It's about time, so I think that's the last one.
Okay.
Last one.
Okay. In a sentence or two, what's the main insight you want listeners to remember from the podcast? Okay. You want to go first?
The main takeaway is that cell therapy is difficult, it's hard, and so partnering with the best in the field, we just believe it enables to reach scale and efficiency by combining essentially the strong suits of every partnering organization.
Yeah.
I think this particular collaboration is a fantastic showcase of how far you can go, how rapidly, when you truly, are aligned on this partnering-based approach.
Yeah, I totally agree, Fred. And the fact that partnerships are important because these are some really challenging problems. We are dealing with patients who are waiting for therapies, and a lot of them are late-stage patients, and so keeping them in the center. Kind of thinking about what solutions can we all brainstorm together from a technology standpoint, from a manufacturing standpoint, from reducing timelines to improving their outcome. I think partnerships have a huge value in that. So from that point of view, we really value the GenScript Multiply Labs partnership, and I hope we can work on future platforms together as well in cell therapy and beyond.
That will be fantastic.
So really looking forward to it.
Wonderful. So thank you very much for being here, Sanket.
Yes.
This has been a great chat and conversation, and looking forward to see you around again.
Yes.
And to continuing and expanding the partnership.
Perfect. Thank you, Fred. Thanks for having me. All right.
Wonderful. Bye.
Bye.
Hello everyone, this is your editor Winston here, and I hope you enjoyed the first episode of Cellmates. We're always on the lookout for new guests to bring to the show, so leave a comment below on who you want to see and what questions you want to ask them. Till next time.